Bismillahirrahmanirrahim. Assalamu alaikum everyone. Myself Sunana Malik, and the topic of my presentation is DNA microarray. These are my group members. Here are the contents and introduction. Microarray technology. The development of microarray technology was enabled by large-scale genome sequencing and advances in surface chemistry. The large-scale genome sequences was allowing for the identification and analysis of thousands of genes, enabling the immobilization of DNA fragments on coated glass, silica, or membrane. This technology has revolutionized the fields of genomics and have been widely used for gene expression, genotyping, and DNA sequencing. Microarray technology has had a significant impact on understanding of biology and the diseases and has led to the numerous breakthroughs in the fields such as cancers, research, genetics, and personalized medicines. What is an array? An array is basically an orderly arrangement of samples that allows for the matching of non-DNA samples and unknown DNA samples. Non-DNA samples are termed as probes, while the unknown DNA samples are termed as targets. The matching is based on the base pairing rules. As we know that adenine always pairs with thymine while the cytosine pairs with the guanine. By exploiting these base pairing rules, microarray enables the researchers to identify the specific DNA sequences, analyze gene expressions, detect genetic variations. Microarray are the powerful tool of microbiology, allowing for high throughput analysis for DNA samples. Principle. The core principle behind the microarray is indeed the hybridization and the probe sequencing single generation. The hybridization between two DNA strands specifically gives the probe sequencing and target sequences. While the signal generation, the strength of the hybridization signal depends upon the number of the paired bases between the probe and the target sequences. This principle allows the researchers to measure the gene expression level and detect the genetic variation. The specificity and the sensitivity of the microarray technology rely on the precise intentions between the probe and the target sequencing, making it a powerful tool for genomic analysis. DNA microarray technology is basically a high throughput method for analyzing the thousands of genes simultaneously, versatile tool for the studying the gene expression, applicable for various research areas. It enables the researchers to analyze the parallel gene expression and to study known and unknown gene, providing the insight into the gene function and the regulation. This technology has revolutionized the field of the genomics, facilitating the discoveries in fields like cancers, research, genetics, and personalized medicines. Each spot on microarray represents a specific gene or sequence corresponding to the genomic DNA fragments, cDNA, PCR products, and chemically synthesized oligonucleotides up to 70 meters. These probes are designed to capture and detect the specific target sequences, allowing researchers to analyze the gene expression, detect the genetic variations, and many more. This is the graphical representation. Principle of DNA microarray technology. The principle of DNA microarray technology relies on the complementary base pairing and the hybridization. Complementary base pairing means the specific binding between the complementary DNA sequences, while the hybridization is the immobilized DNA base probes captured labeled target sequences. These principles enables the researchers to detect and quantify the specific DNA sequences, making microarray a powerful tool for gene expression analysis and the genomic research. Sample preparation and labeling. It Firstly, the RNA is isolated. RNA isolation is the goal, because the RNA isolation include messenger RNA from a sample. This RNA should ideally represent the gene expression profile and should have to be quantitative. This isolated RNA can then be used for various downstream applications such as gene expression analysis using the techniques like microarrays and the RNA sequencing, understanding the biological processes and identify the potential biomarkers or therapeutic targets. This is the picture that is showing that how do we do this process by isolating RNA. Hybridization. Hybridization is is done into two steps. cDNA samples are collected, which are labeled with one dye, and control cDNA samples, which are labeled with another dye. These isolated cDNA samples are then mixed then to hybridize to the microarray, allowing researchers to compare gene expression levels between the samples and the control. Next step is the washing. After hybridization, microarray is washed to remove a non-specifically bound label cDNA or cRNA. Reduced background noise. This step helps ensures that especially bound label micromolecules remains, allowing for more accurate detection or quantification for gene expression. Image acquisition and data analysis. After hybridization and washing, the microarray is scanned to the acquired images and to analyze the data. Acquired images captures the fluorescent signals from the labeled cDNA or cRNA as it is showing in the picture, and analyze data, extracting the intensity values to normalizing and comparing gene expression levels between the samples. This step involves using the specialized software to quantify gene expression, identify differently expressed genes, and interrupt the biological significance. This resulting data can provide the insight into the gene function regulation and the potential therapeutic targets. Microarray slide is dried and then scanned. Then the the scan generates the image that are then analyzed to measure the fluorescence intensity, comparing the gene expression. Hybridization signals. When the labeled targets hybridize to the probe on the microarray, it produces the fluorescence emissions, and these the signal intensity provides the proportional to the amount of target bound to the probes. These emissions are detected during the scanning process, allowing researchers to quantify the gene expression level. Types of DNA microarray. The glass DNA microarray, and the other is high density oligonucleotide. Glass DNA microarray involves the microspotting of the prefabricated cDNA fragments onto the glass slide. This approach allows to the customization and the gene expression analysis. Customization is that researchers can create the array with specific cDNA probes, while the gene expression analysis enabling the study of thousands of genes simultaneously. Glass cDNA microarrays are the popular choice for the various applications, including the gene expression profiling and cancer research. While the high density oligonucleotide microarray, also known as CHIPS, these microarray involves in situ oligonucleotide synthesis, direct synthesis of the oligonucleotide on an array surface, high density thousands to the millions of the probes per array. This technology enables the high throughput analysis and the precise measurement of transcript levels of and genetic variations. And examples includes the affirmatics, gene chips, and the other commercial platforms. Glass cDNA microarrays. Glass cDNA microarrays was indeed firstly proposed by Patrick Brown and his colleagues at Stanford University. First type of DNA microarray technology developed for the gene expression analysis. These microarrays are created using the robotic printing and high density spotting, allowing for the thousands of genes to be analyzed simultaneously. This technology has paved the way for various applications in genomics and biomedical researches. Advantages and disadvantages of microarrays. Some benefits are displayed here as affordability, accessibility, and data capture. Advantages contains affordability, as a glass cDNA microarray are relatively affordable with the lower cost, while they do not require any specific equipment for use, and hybridization does not require special equipment. Their data can be captured or carried out by using simple experiment that is very often already available in the laboratory. Disadvantages of glass cDNA microarrays are intensive labor requirement for synthesizing, purifying, and storing data DNA solutions before microarray fabrication. They may be hybridized to spot the design and detect the transcript from a different gene. In situ oligonucleotide array format. This format involves the direct synthesis and the high density arrays. The direct synthesis of oligonucleotides on the array surface and the high density arrays thousands of the millions of probes per array. Examples include the affirmatics gene chips using the photolithography for cytosynthesis. This format enables high throughput analysis of gene expression, genotyping, and more precise measurement for the transcript level of genetic variation. High density of oligonucleotide arrays. There are some features which they represent, and these are the in situ synthesis and high probe density. Applications include gene expression analysis, mutation detection, and SNP analysis. Advantages of in situ oligonucleotide array format includes speed, specificity, reproductivity. Disadvantages of in situ oligonucleotide format. In situ oligonucleotide array formats tend to have the expensive specialized equipment is required to carry the hybridization, staining and labeling, washing and quantification process. Short sequences used on the array have decreased the sensitivity binding composed compared to the glass cDNA microarrays. Application of microarray technology. Applications of microarray technology have various applications in gene expression analysis, genetic diagnostic, cancer research, pharmacogenomics, and pathogen detection. These applications enable researchers to clinicals to better understand complex biological processes and develop the personalized medicines approaches. Microarray, a gene expression profiling tool. DNA microarray technology helps the scientist to find out that which genes are active in a cell, when they are active, where they are in a body, and they are active and how much they are active. In simple terms, it's just like checking the list of genes which are seen to ones who are working in a particular sample or tissue and how strongly they are working. This helps in understanding that how cells behave in different conditions, like in healthy versus in diseased. Microarray analysis checks that uh, by doing this they can understand that how genes are controlled and what their role are playing in the body, such as in growth, diseases or in other functions. Microarray as a competitive genomic tool. Microarray technology is used to compare the DNA in of different organisms of to find out the small changes like mutations or single letter differences in SNPs in the genetic code. In microbiology, it helps the scientists to spot the genetic differences between bacteria even if they are very similar. This is a very useful tool for understanding that how do we relate or why one make can cause a disease while the other one cannot. Disease diagnosis. Before microarray, the cancers or the other diseases were grouped based on the organ they started in, like in lungs, breast, or liver. Now, with the microarray technology, scientists can look at the activity of the genes in cancer cells. This helps them to better understand and classify cancers based on how the genes behave, not just where the tumor started. In simple words, the microarray helps to find the more detailed type of cancer by checking which genes are turned on and which are off in their tumor cells. Drug discovery. Comparing the genes of disease cells with a normal cell type, scientists find out that what changes will happen in the disease gene. This comparison helps to identify what kind of the proteins is the disease genes are making and what kind of the medicine is should have to be promoted to stop the wrong protein to be caused. The disease and the finding possible, this helps them to find the disease and the and finding them for the possible treatments. Toxicological researches. Microarray technology is a powerful tool which helps the scientists to study the toxins effect cells, whether these effects are the passed on the future generation. This area of the study is called taxonomics, which looks how the exposure of harmful substances causes the changes in gene activity. Microarray allows the researchers to study the thousands of genes at the same time while helping them to understand which genes are more or less active than toxins exposure. This gives a clear picture of how the organism responds at the different genetic levels. These are the conclusions. Microarray is a recently developed functional genomics technology that has powerful applications in a wide array of biological, medical sciences, genetic biotechnology, environmental sciences, and industries. Since many universities, researcher institute, and the core facilities have established microarray laboratories, microarray have become an easily accessible and widely used technology for investigating the biological systems. Thank you so much.